Very excited by the lineup of speakers, and today we are gonna have the first one, who is Professor Martine Ben Amar from the Laboratoire de Physique at École Normale Supérieure and the Faculty of Medicine at Sorbonne Université. So she works on many aspects of applied mathematics, in particular growth, shape, dynamics, tumor modeling, biomechanics of animals, and morphology of animals and vegetables, many things. She leads a research group in physics and mechanics of soft matter, and she was awarded the Le Prix Huy Duong Bui in 2018. She has many interesting. She does many interesting things, and it's a great pleasure to have her here.
Okay, thank you very much. Thank you. Let me know when I begin. I begin? Okay, I begin. So thank you for the invitation. I was rather impressed for this invitation, and especially to be the first one. I have a problem when I prepare my talk, because I do as usual. So I explain mechanics. I hope that it's not too far from your field, and okay, I will try to answer questions, but at the very end. So I focus on thin lining tissue because it corresponds to what I have done recently with my PhD student. And okay, so I begin. So I will speak of the basis of morpho-elasticity, of lining tissue, thin and not thin, by the way. Morpho-elasticity means that we are interested in shapes mostly, and we consider that because we focus on living tissues, which are stuffed, elasticity is something really important, but also growth is important, and especially volumetric growth. So this coupling between finite elasticity and volumetric growth, if we succeed to treat this correctly, is supposed to give you the growth, the shape of the body as when it grows. The approach is continuous. It means that, of course, we cannot too much go at the level of the cell, or with some difficulty. Let us say that we are above the level of the cell, but it works for few cells. When you have a collection of cells, it works. And of course, it is well suited for embryos, organs, plant, and also if you want to explain pathology like wound healing and of course solid tumor. This model, so it is well suited. It requires that growth is very slow. And this is the main hypothesis. Usually for organs, embryos, and so on, it is really slow. For tumors, the time scale is the year, so it's really slow. And especially it is slow compared to any physical time scale, like dissipation, even remodeling. I will show you a case of remodeling, which is slow, but okay. In principle, growth is much slower than any other time in our problem. So it means that we do not have a dynamical equation to solve. So this is nice. We assume that elasticity adapts immediately. So this is the hypothesis, and as soon as you assume that, you understand that what we have to do is to minimize the elastic energy, because in physics, we are always looking for minimum of energy, which indicates equilibrium. So now, what are the tool? The mathematical tool. So the mathematical tool is mapping between two configurations. the configuration of at time t equals 0, let us say, when the growth process begins. So you have a body with a shape, which is here. I don't know if you see my pointer.
Yes, we do see your pointer.
You see the pointer. When I do this, you see.
Yes.
So you have initial shape. And after some time, you arrive to this shape. And of course, you need a tool to say that this point, which is red, comes here. And this tool is a mapping, what we call a mapping, which is in principle in 3 dimensions. Although after I will focus to 2D, but for the moment it's 3. And as soon as you have a mapping, which is here, which is a vector, in fact, what you are interested is the gradient of this deformation, which is given by f, which is a tensor. This is the gradient of chi. And the main assumption in morpho-elasticity is this law, which has been written a very long time ago, which is a law which is also valid in plasticity, in other words, in other problem of mechanics, which tell you that simply the geometric deformation that you see when you are interested in shape is something that you see. In fact, this F is simply the produce of an elastic tensor and a growth tensor. And this growth tensor, a priori, is given by the biology, which tell you that at time t, that I consider for the observation, in fact, the amount of growth around some small volume is G, this is the, so is G, is big J, and J we will define later. So it is simple, it seems simple. There is a graph which is standard to explain this. In fact, I don't like too much this graph now because I noticed that it is a graph to explain the origin of strain and strain, yes, but we are never in such configuration. The idea was that this G that we introduce as a growth tensor is a mathematical object which can have problem in itself, you know, giving a strange shape, and elasticity is here to recover a good shape. But in fact, we are reasonable people, and it's not useful to explain the origin of the strain and stress. Okay, but anyway, the model is from Rodriguez, is here, and in plasticity is another. Easily. So, in living tissue, what is the main origin of stress? In fact, it is as soon as the growth tensor has some symmetry, but for some reason, the deformation doesn't follow this symmetry. And this is the origin of the bioinstability, which explains many shapes that I do not consider here, but I mention. And the other origin of stresses is the existence of layers. And this is true for in our body. As soon as you have an epithelium, the epithelium is attached to the extracellular matrix. Sometimes you have a basement membrane, and so on. So it means that you have what you think as a layer, it's multi-layers. These layers are connected, but they want to have a different growth. And it is automatically a source of strain and stress, like, and which at the origin of distortion. This happens for our fingerprints, the brain cortex, and the villi. And simply, if you want to know, for example, when fingerprint appear on a fetus for human, in fact, you get these fingerprints after 4 months post-fertilization. So a fetus has no fingerprints, except when he has 4 months. And this is the same for the cortex of our brain, and it is the same for the villi of the intestine. So here you have a specimen of villi. It is villi of turkey before hatching of the egg, of course. And you have here, and you have simulation. In this picture, I show a brain, and this brain seems really true, but it is not true. It is made by gel swelling, and this experiment has been done in Harvard in the group of Evan. And if you apply the law of elasticity, as you see, this is what we have done for human, quite without parameters, without fitting parameters, you can represent by this theory, in fact, the girification index, which is an indicator of the folding of the brain. And this is data which we are known from autopsy. I don't know how they know this, because it is for fetus. Okay. So it means that we are able, with simple law, with and simple geometric consideration, to justify this theory of morpho-elasticity. And here I show you the main equation. Do not worry, I will not use it, but it's simply to say that as soon as you have the elastic tensor, you have the Cauchy stress, which is written here. So you have the stress. So the stress is related to the strain in elasticity. And we use incompressibility, which is very well checked for living tissues. Okay. You have also, you can add forcing, for example, gravity, if you want. Gravity you put here. If gravity is important, it's not always, but if it is, you add gravity, and you have boundary condition. And if you are able to solve this system, in fact, you have the shape of the body, the instability of shape of the body, and so on. And the main density of elastic energy we use is the Neo-Kean one, but if you are more rigorous and you say, oh, but there is fibers, it's not a big deal. There is plenty of model in the mechanics to adapt to a specific case. Okay. So now I come back to my topics. I would like to speak because now we are convinced that with volumetric growth and because of layers and so on, in fact, each time we have growth, we have stress. But it's not so true and not always true. And this question, first I have seen, in fact, it was a question of a group of Tianjin and Hong Kong. I'm sure my PhD students are laughing at the moment when I say that, but okay. By Vividai, this is the mechanician, he asked the following question: is it possible that to have volumetric growth, and what it is with G tensor and no stress? So, okay. So we decide to apply, so he has a formalism to get this, so it seems to be possible. But we notice that he is always using a circle or sphere, and we were more interested in leaves. So fresh leaves. So why leaves? I will explain you very soon. So first of all, I think in America it must be exactly the same, but you have in Europe, at least in all the laboratories, all administration, all hotels, you have this plant, and this plant is here, which is here, and this is a plant which has holes, and which is all the leaves are planar, and really planar, except the very big one, because I think the gravity is, but they are really planar. And first of all the elasticity of soft matter is not so obvious to imagine. And we say, and okay, if I have a young leaf, it is really planar. And if you try to measure the stress, we cannot measure, but we can demonstrate the existence of stress on this leaf by doing two cuts at right angle. You do two cuts at right angle. So it's very easy. You have a knife and you do two cuts and there is no stress. So obviously, a fresh leaf satisfies the condition of growth without stress. And it's really strange, first of all, because of the hole. The hole is very... And another thing which is strange is how the leaf appear. And here, and in fact, the leaf appear in a stem, in this stem, which is here, which breaks at some moment, and it appears like a cigarette paper, the cigarette paper of the old times, more or less, completely like this, because it was before in this stem, and after it opens, so you have 2 days, and finally, in 2 or 3 weeks, we have a planar leaf. So this is a very good example of elasticity in the sense that you can explain very well to a student what it is remodeling, because after 2 or 3 weeks, you get a planar leaf, but really planar, yellow, nice. After it change, the color change. And what we notice is that most of the time, the holes pre-exist. So it means that the holes are in the stem. And the biologists who were interested in this plant they are convinced that the origin of the holes is genetic. So genetic is nice because it's not our problem. We do not have to interpret holes, so it's nice. And but what it is rather strange also is that okay, it's very popular, but the word which is fenestration, fenestration for the leaf, in fact, is a very rare event in the world, in the botanic world. It means that either you are faced to a monstera or it's not a monstera and there is no hole. So, and no pre-stress. So, okay, if you have no stress, what happens? So you have this tensor, this geometric tensor that I call F, and you have this G. It means that, okay, no strain, no stress, and the elastic tensor is identity, so F equals G. But another equation is G. Another equation is what it is with G, in fact, for this leaf, which is rather specific, but for any leaf. It's obvious that the shape of the hole reminds us of a problem in hydrodynamics, so in conformal mapping. And I was quite sure that there is something similar to hydrodynamics, viscous fingering, Safman-Taylor viscous fingering, and so on. And in fact, it's not difficult to show. So I will be fast because perhaps it's too much math. But when you have two planar curves, which are closed, and not too much tortuous, two planar curves only, in fact, you can find a mapping of the outer contour, of both outer contour, which is a complex function, an analytic holomorphic function. And in addition, the inner part is mapped to the inner part of the first one is mapped to the inner part of the second one. So in fact, looking simply at the contour, you have a mapping. If you use this mapping, which concern only contour, and you say that in fact, in addition to that, I say that the deformation follow this mapping, you are safe. It means that because of the Riemann theorem, okay, you check this and this, you have the determinant of F. It means that the growth at a point X and Y is given simply by this. It's always positive. So, and in addition, you do not have elasticity. So now I do something more strong. I say, in fact, the function chi I have defined at the beginning in 2D, I say it's given by a conformal map. In fact, it is this function C. So it is perhaps a little bit difficult to understand, but mathematically is okay. The things become easy if you introduce this idea which concern growing object without stress. So if we do that, can we explain the shape of leaves? Yes, very easily. We begin by an ellipse. This is the shape of an ellipse in complex analysis. We use the coordinate, curvilinear coordinate of the ellipse, given by that. We do a small expansion, and here I have only three components. And what I get the leaf I want. So this is marvelous. And here you have different specimens with different tip. And we have the shape we want to represent. In addition to that, you have the amount of growth in each part of the leaf to represent the things. Okay. So, okay, but there is no hole. So now I want holes. I want to represent a shape with holes. It's not complicated. It's not complicated too much because we need complex. The complex analysis has been developed a lot in viscous fingering by mathematicians. And the case of a bubble in a Eléchaud flow and a traveling bubble in a period, so in a rectangular, has been solved without surface tension, but we do not put surface tension in elasticity, so that's nice. And for the paper we work with, it is this paper. But okay, we can find many solutions on such problem. I simply want to say that there are specialists on these topics, and I have cited some of them. But Tony DeLillo in the US, I do not remember where, but with a specialist of very strange contour and how to recover easily and to check to obtain the function chi. Sally Tanvir, it is for the Safman-Taylor problem, bubbles in Eléchaud flow. Linda Cummins, who is here, and also Darren Crowdy, Linda in New Jersey, and Darren in London. And this is exactly what we do. We use this bibliography, and how to get, we have the rectangular with a hole, and we put this hole into a sector of our leaf where we want the hole. Because of course we can separate the global shape into sectors, and we reproduce holes, we reproduce fingers, and we reproduce fingers close to a hole. So because you have a variety of Monstera who have holes and fingers simultaneously. So this is what I want to say. So I want to say also the idea to use in 2D conformal mapping to represent living tissue, living object is not new, of course. Because, okay, here you recognize perhaps the book, some pages of the books of Darcy Thompson on growth and form, more than 1 century ago, a book written by a biologist, but who obviously like very much for mathematics. The word complex function, conformal map doesn't exist in the book. I checked, but I have not seen. Anyway, it's really quite a conformal map. And also there is a experimental shape by Yareski, Boudaoud, and Boris Schreman, and Karen Halim for the mathematical representation on tobacco leaf and petunia. So I do not remember what it is, the tobacco or the petunia, and they check that the growth follow some kind of complex conformal map. But what I would like to say, what we have done is to prove that in fact it is a way, it is really a way to suppress elasticity, so to suppress, not to avoid elasticity, but to eliminate the elastic stress if we follow conformal mapping. So now I continue. I think, yes, I have time. So I continue. In fact, I cannot follow conformal mapping completely. In this case, even if you have a thin sample, and here I have a thin sample, which is a plate, you have a distortion. So this is, you have a distortion. And I assume some leaves, have distortion. Even fresh, okay, they buckle. Okay, so they buckle, it means that they refuse to follow conformal mapping, so which is bad, but okay. So I want to say the Vodkarman equation also has more than 1 century. I have forgotten to write the date, but it's 1907. Here, so it's very popular, and it's extremely popular in physics, much more than in mechanics for some mysterious reason for me, but okay, very popular in physics. So many also paper, especially in the community of nonlinear physics, with the Foucault-Karman equation for plates, and of course with a physical viewpoint. So I show you the, so when it is valid when the thickness of the plate is very thin compared to the dimension, horizontal dimension, but it allows some non-linearities of the distortion. It means that the deviation zeta, which is not here, but which must be here, in fact, it is much bigger than H and smaller than big L, which is the size here, so intermediate. And what happens, and I will insist on that perhaps, is that if we have full homogeneity and a unique plate. In fact, there is a zone which is called the neutral surface, which is here, which is in the middle, in this case, where one part of the upper part, in this case, it is in tension, while it is in compression below. So you have a surface of separation in the middle. Okay, it's important for the following. So I have written the equation simply to show you. So first of all, this really nasty equation for Forda, nonlinear and coupled. Here you have the Young modulus, because this equation has been written 1 century ago, and nobody speak about finite elasticity at that time. The thickness. So two equations. The things which is important is, in fact, these two equations concern the mean curvature obtained after distortion and the Gaussian curvature. Mean is, because here you have a delta square, but okay, it's delta of delta, so mean curvature and the Gaussian. So it's a way to get really the geometry of the distortion by operator, by the mean curvature and Gaussian curvature. So it's not astonishing in the geometry of surface, but okay, we recover. And what happens if you have gross and finite elasticity? But what happens? You add terms, obviously. So if you have at exterior pressure, I put here vertical. Okay, you can forget this if you want. But anyway, if you have gross, it turns out you have more terms. And we have calculated in our group a long time ago, more than 10 years ago, what are these terms? Okay. So you have a force mean curvature, which is written here, and a forced Gaussian curvature. It means that, in fact, if you do not want to have stress, again, you have to follow the growth geometry. And if you do not follow exactly, you get stress. So this is the same idea as before, exactly the same idea. Okay, so it's very bad equation, and by the way, it has been done a long time ago, so we can forget, except that I was contacted by a friend from Marseille who has a problem with Drosophila imaginal disc. So, and but the nice thing, so his problem is much more complicated, but the nice thing is that is roughly in one dimension. And in one dimension, you have plenty of simplification. So it means the equations simplify. Here, this one means simply that sigma, the stress in plane, is a constant. This one simplify also, so many simplification. So we can, in 1D, we can forget this difficulty. And I come back to the biology, if you want. I remind you that Drosophila, okay, it's a model system for embryogenesis. People are interested in the imaginal disc. It means the location on a wing or on a leg, it's possible also, of the stem cell of a larva. It's a larva. Donc a larva. So the stem cells are in some I will show you the imaginal disc just after. The imaginal disc is not fully a plate. It is mostly a pocket which is bent, a pocket so on top and below, and which is attached to the remaining of the ring. And the idea the biologists have, there's biologists who spend a lot, biophysicists, not biologists, but who have spent a lot of time, more than 10 years on the Drosophila. It is to use a mutant, and mutant, of course, have defect. Okay, so, and to observe what happens on the imaginary disk. So this is the imaginary disk of Drosophila. This is the shape is a pear. So we will forget the pear and we will imagine simply a stripe like this, something more simple. And because of genetic mutation that we succeed to induce to the Drosophila, they have a zone of defect. So what kind of defect is another mystery, but this is a defect. And what happens is that the pocket, instead to be like this, present is like this. So some kind of small collapse, I will say collapse, and like this. So to give you an idea, this is the dimension, 30 micron. It requires sophisticated optics to get that, very difficult, especially because the tissue are more or less transparent. Okay, they succeed to have fluorescence, but the difficulty is the optics, really the optics. And this is what they get, and they want to understand what's the origin of that. So again, the scheme of an epithelium, standard epithelium, it is one layer in this case. So it's not the epidermis. It's not. It's more complicated than our epidermis. Only one layer, and they have a substrate, which is here, which is the extracellular matrix, and so 2 layers, and you have also a compression which is due to adjacent tissue of the wing, which is here. Okay, so if you consider this, it's complicated, obviously, but perhaps the advantage is that these 2 layers they are thin. So perhaps we can use von Karman equation, and perhaps we can use this von Karman equation for getting in 1D. This is what we will do. This is what we have done more exactly. And this is the same von Karman equation. So the second one I show you, it's simple. It means that the compressive stress coming from the tissue is a constant. So we do not know this constant, but there is a constant. And it's obvious because the imaginary disk buckle. So no way. You have this N0. This is the standard 1D von Karman equation for the vertical deviation, but you have gross contribution. And the gross contribution, if everything is regular, are constant. But in this case, it is not regular because you have this defect, this mutant. And this is the expression, so it's rather awful, but you have the gross tensor, which is here, the component of the gross tensor, which is here, 2 components, two here. Okay, so you have two. Okay, so I am too fast. So what you have standard equation, but you have this, and these terms are due to growth. Okay. So I skip this because it's perhaps a little bit technical, and I am not on time. So if I have a defect, as you see in my equation, I have a right-hand side. This right-hand side exists if I have growth, and this is the growth component, which is written here in 1D. So it's 1D in 1D. And if I want to have a significant signal, I take a defect, and I take a defect which is localized, because I have to derive it 2 times, so it is more efficient. And I put the defect, I don't know where, it doesn't seem regular, so I put the defect not in the middle necessarily. Here it's not in the middle. I have a width of a defect, and I have an amplitude for a defect, and you make simulation, and of course you see a backlink. It's clear with this equation, which is absolutely not complicated. So now, if we compare to the experimental data, remember that the scale is 30 microns, so here it's several microns. The problem is that I do not know what happens before because it's a mutant. I do not know what happens when he is healthy. He is never healthy, he has a defect. But okay, he has a defect. But I have to assume the shape, and I take the shape of the asymptotic, and this is on behalf two mutants, so two age different, and you see that we represent very well, in fact, the deformation of the imaginary disk. So it's simply to show you that morpho-elasticity works only at small scale, and it's the advantage is that it very few parameters enter in the modeling. We have a way to vary the parameters. We have good software in case of complicated situation. Of course, in this problem, the experimentalists expect that I explain them what the mutation concern. I cannot tell them if the mutation concern growth or if it concerns the structure, because the defect can be a defect of structure in this case, or a defect of growth. Okay, so the paper was done by my 2 PhD students here, and by my colleague, the experiment, the group of Marseille, Loïc Le Goff. And for the, I don't know if I was very precise, but for the leaf, the work was done by Anna Dai, my Chinese PhD student, and I know she listened me at the moment, so I want to mention also. So I am done on time, I think. So thank you. I hope I can go back to and see the face. Yes, I can.
So
Not
let's
everybody,
thank,
but some of you.
yeah, let's thank Professor Ben Amar for a great talk. So now we have time for questions. I'm gonna just first of all post a job advertisement. We're gonna have many jobs open between the labs of Katrina and myself. You can give a look at that. There is one already there. You're gonna check whatever you want, advertise it. Feel free to, if you're interested, discuss that. Well, also questions. I have some, but Kathleen, please.
Okay, so I just wondered, you mentioned how uncommon it is to have fenestration in the botanical world, in the natural environment. And the species you were looking at also had no stress in the leaves. And I'm wondering if the two might be related. Is it also uncommon to have no stress? Like, could the fenestrations be a mechanism to remove stress during growth?
No, I do not think so. When a leaf is really planar, really, because in leaves, the structure of the leaf, the transversal structure of the leaf, if you cut the leaf, is complicated. It's really complicated. And I will say naively, if you believe what it is written in this literature of biomechanics, automatically you have the feeling there is plenty of stress. Because of this structure and because each wants to grow differently. So that's it, it's enough. And in fact, when we begin this study, we focus immediately on this one because I think it was too much fun. But and I say we buy a cutter simply and we cut and we observe that there is no stress. And for me, after so many years, of calculus of stress and strain in growth, I was really amazed. And this is the question I was really interested. Not so much of the whole, but after I see this relation with conformal mapping, which works marvelously, that you can represent any shapes. And of course, I apply to this one because it's, as I explained, it is really strange. And it is true that there is not so much holes in leaves except family of the Monstera. So I don't know why, but it's like this. Okay, so I speak a lot on the holes because we can prove also and show the powerful of conformal mapping in this case. That's it. No, it's only for fun, but no, we were really surprised that, and it opened us, opportunity to do more with the von Karman equation. And we will do more after. I cannot say what I plan to do just after, but I have more projects because I eliminate simply one equation of von Karman, which is really nasty. So we will do progress, I'm sure, in the future with this idea of conformal mapping.
Awesome. So related to that, actually, I can just ask something, right? So is there restricting to conformal transformation is too much in 3D. So is there something else that one can do? Like so conformal works very well in 2D. Is there something that one can do in 3D? Of course, not conformal, but something else.
So this is a question also, so you are specialist here on this question of extending conformal map. Linda can answer this. Okay, so when I do this, I have done very few times this seminar because it's really new. It's really new. Okay, you have the idea, but there is construction of conformal mapping in 4 dimensions. So it means you have 2 dimension.
Like the Panitz operator or Q curvature, that type of things, correct?
Ouais. So, but I don't know. I don't know if it will work. And sometimes you may think also to axial symmetry, to do something more or less, axial symmetry. Okay, it's not our immediate project anyway. And also, whereas you can notice that I always change projects. So also, no, there is things to do. I'm sure with other situation where we can use this idea of conformal mapping as 0 order.
I mean, there are some, just to add to what Martine said, there are some applications where, for example, you might have a 3-dimensional geometry that varies only weakly in one direction. So I've done some work on evolution of viscous fibers, in fact, related to fiber drawing. And because the fiber shape varies only weakly in the direction that you're drawing it out, you can essentially use conformal mapping methods to describe the shape in the plane of the fiber, but your conformal mapping will contain axial distance almost as a parametric dependence. So you get a conformal mapping that depends
on distance along the axis of the fiber in this particular application and on time t, but the actual cross-sectional shape is defined by a complex analytic function.
Ouais. So, yes,
Right, something like conformal plus a perturbation which involves a third dimension, something like that. So, other questions? Well, the other thing is, so you mentioned that you stop. Well, actually, basically, the first comment that you made, you stop at you cannot reach the set level. So, how to do that? Like, do you need to just completely change the theory? Yeah.
How to do that? First of all, if you go at the cell level, you begin to have plenty of problems with the biophysicist. And in France, the community is very strong, very tough, and you begin to have problems. So it's a reason not to go below. No, because they have developed their own formalism and, the vertex model, all these models that they use. And it's so I was very happy when Loïc Le Goff contacted me, because he comes from this community and he was so used with the vertex model. He wants me to do vertex model too, and so on. And this approach, but okay, when he see that it works well, he was happy. No. You have so there are plenty of things which are done at the level of the cells. Plenty. At some moment, you have some complexity. So if you are at the level of the cell, you can treat many important things at the level of the cell, but you cannot go to macroscopic scale, or it is very difficult. So I think we must adapt the tool. If you consider, I don't know, a plane in the sky, you do not go back to quantum mechanics or quark, the physics of quark. Huh? If you do that, it will never, it will not fly. So you have to adapt the tools.
Thank you. Other questions? We have time.
Just a question, Martine, about your last slide here. So here you're comparing a 1D model with real 3D observations. Is that right?
Yes. 3D observation is okay. It's they observe what they can. You remember that they want to keep the embryo alive, because all the biophysicists, they want to work with alive. So the embryo must remain alive. So it means a light which is with a small number of photons, as they explained me, the smallest number of photons available, that more or less everything is transparent. And so not too much. Okay, so we have many problems. But when I see what they are able to give me as information, I say, okay, 1D will be enough. 1D is enough. And as I explain, I do not know what's the problem of this mutant. Is it the epidermis? Is it the CM? Is it the growth? Is it the young modulus? What the problem? I don't know. And they cannot tell me what's happened to the mutant, except that they can visualize us. Okay. So it's not really necessary. Also, we have to adapt our work compared to what they can give us.
Great. Well, if there aren't any more questions, thank you so much, Professor Ben Amar, for that great seminar. It was very, very interesting, even for those of us like me who's less mathy, but really enjoyed it. So thank you so much for a fantastic kickoff to the new season of MaLaMoBi seminars. I think Alessandro also posted the link for the seminar page for anyone who wants to see some of the other speakers. That's growing constantly. So hopefully we'll see you again.
Also, I don't think I write my email, but if I don't know, students have questions and so on, we can send me an email. I will answer.
Great.
Okay.
Thank you so much.
Thank you.